Hello, my name is Tony Pridmore. I'm Professor of Computer Science at the University of Nottingham, and in the next 20 minutes or so I'd like to tell you a bit about some of the work we've done over the past few years aimed at root phenotyping, particularly using deep machine learning. When we started in this area around 15 years ago, the world was very different. Back then, people in my field would use classic techniques of image analysis and image processing, combine them, manipulate them, modify them a little bit to produce application programs that were hard-coded, could be downloaded and run on people's machines. And that's what we did. We used particle filtering level sets, network snakes, things that had been around for a little while. We tuned them, modified them, and we produced a set of tools for analyzing various types of images of roots. So this was all hard-coded. We took our knowledge of roots and images and we put it into code explicitly. And if we had to change something, if a tool was required to run on a very different sort of image, we had to go back and we had to recode it. Our goal back then was to produce fully automatic tools, and sometimes we got close, okay? But the nature of the technology and the difficulty of the problems meant that most of the time we ended up needing some user input. So after a while we kind of changed our goal and moved away from trying to do automatic stuff to saying we need minimal human interaction. There's probably going to be some, but we want to keep it to a minimum. So one example of this is our RootNow system. This takes images of roots grown on blue growth paper and pulls out the root architecture and stores it in a form known as RSML, a root system markup language. First thing we need to do in that is detect the root tips, and there are a variety of feature detection methods out there that could be used for this. Unfortunately, as you can see on the figure on the left, although they found the root tips, all of these methods tended to give too many false positives. The root tips are actually more complicated than you might think, and so we needed to go to the picture on the right, and we needed to have the users help the system. We still ran the feature detector to actually do the precise localization, but we made users click near the tips and then we would use their input to select which of the features were the good ones. Okay. 7, 8, 10 years ago now, deep machine learning appeared in computer vision and made quite a huge change to the way people work. So deep learning methods do not use pre-computed features. This is the whole point of them. They are neural net type systems usually, and as well as doing what classic neural networks do, which is learning how to map their input to their output to do some task, deep machine learning methods also learn the features that you need to be able to do the task. So if you look at these two images here, one of them on the left has a root tip in, you can perhaps just see the white tip. The other one on the right is a crossover. Hard coding something to tell the difference between those two is phenomenally difficult. Yeah? It will appear in all sorts of different ways. These features will appear in different ways in different images. Really, really hard to figure out exactly what to do. For a convolutional neural network, it's not so hard. You give it lots and lots of examples, yeah? And the system can learn, as long as you give it the right input, what a root tip looks like and be able to solve this problem. So the deep machine learning approach tends to give better performance. It tends to use features that are quite similar but a bit different to the ones that people would use if they were hard coding. But it needs to be trained. You have to have input images and the desired output. Yeah? And that means that these images have to be annotated. So what we found was our first generation systems like RootNav and so on actually became annotation tools, right? And we generated quite a lot of data using them, and in fact biologists using those tools previously had already generated a lot of data for us. So there are a whole set of deep machine learning methods and networks out there. Sometimes you can apply them to your problem and just solve it directly, but not always. I'm going to show you a few examples now of these types of things in our work. So one very common type of neural network out there is the encoder-decoder network. And it was originally developed for things like this, for segmenting an image. So you see a road image on the left and a segmentation on the right in which different parts of the scene are labelled in different colours. Right? Semantic segmentation means every pixel in the image is classed as in some way, classified. So the pinky ones in the middle are road, yeah? And the blue ones are pavement and so on. The way that this is done in practice is that someone has to take a whole load of images of road scenes and colour them in, right? Provide ground truth, show the system exactly what the segmentation output should be. The neural net then learns to map the input image to a new image in which each pixel is hopefully properly labelled. You see lots of diagrams like this in the neural network world. What convolutional neural networks do in general is they begin by applying a bunch of filters to the image. They then shrink the image down, apply some more filters, shrink it, apply some more filters. The effect is that effectively it produces a set of filters that are increasingly large. Yeah, the relative size of the filter to the image increases as you go in. And you end up with a very compact, in the middle of that diagram, representation of the image in terms of the output of a bunch of filters. What you can then do is reverse that process, upscale the image, expand it, fill in the gaps successively until you get an output that's the same size as the input. And if your system has been given the right amount of data and has learnt it properly, it will have learnt a mapping from the input image to the output segmentation. So we took this idea and applied it to RootNav to produce RootNav2. This is a schematic of our network. It's pretty much the same as a standard encoder-decoder. It takes images of the root grown on blue paper, and it produces an image back, and it produces an output image in which each pixel is labelled by what it is. Okay? It picks up root background, root tips, the seed, and it even classifies the roots as being primary root or lateral root. It's a fairly straightforward application of an existing technique to a new problem, but it was the first one to do it. So here's some of our output. We have something here, a neural net that provides automatic segmentation of roots, tips, seed locations, and so on. Yeah? And you can see the input image on the left and the output image on the right for a variety of different things. To do this, we had to train the network as well as design the architecture of it. And training is something that needs quite a lot of attention and have quite a huge effect on the performance that you get, the different ways that you go about doing the training. So we designed the network. We initially trained it on around 3,000 images of wheat, an output that we got from the previous RootNav. And we got some pretty good results on wheat. We then wanted to use it to look at Arabidopsis seedlings grown on the agar plates that you see in the middle of this slide. One of the very first things we did was something which addressed that problem. So we had data from another of our earlier systems that provided training data to it. Because the network had learned to do wheat and the contents of the network need only be changed a little to map it to Arabidopsis, because it's essentially the same problem in a slightly different world, we were able to do this with comparatively few images, so just under 300. We then transferred this again to rapeseed on the right with only 120 images. So this is quite an important feature of these sorts of technologies. You do need a fair amount of data to train them, but if you have a set of data in one domain and you train it up to be good in that, you will get your network close to what it's needed to do in a related domain. Yeah, perhaps a different image, perhaps a different species, and so on. And then a comparatively small amount of data will be able to transfer what you've learned about wheat into Arabidopsis and then into rapeseed. So this is the final system we ended up with. RootNav produces RSML output, as does RootNav 2.0. So in RootNav 2.0, the images are input. There is a convolutional neural network. The two boxes onto the right, heat map regression and semantic segmentation, are simply indicating that it detects the points, the tips of the roots, as well as segmenting the roots from background. We then have the same kind of post-processing and production of RSML that we had in the original RootNav. So this is effectively RootNav with a convolutional neural network component added, and the old segmentation has been replaced by something that's been learned. The nice thing about this is that this is now fully automatic. Okay? The ability to learn how to do this initial feature detection task that obviously affects all the later stages has meant that we can provide a tool that's completely automatic, which is kind of good. So that's an example of using existing tools embedded in perhaps traditional methods. Sometimes they're used entirely by themselves. Yeah. Sometimes, though, it's not so easy to find an existing network and training scheme that will work for the problem that you have. So another problem we addressed some years ago was segmenting X-ray images of roots grown in soil to pull out the root structure so that we could again produce an RSML description of it. And this is a much harder problem. I think you only have to look at the images to see that. If you look at these images in detail and look where the roots are, which you can see them by eye with a little bit of squinting, the grey level distributions of the soil, water and air all overlap. So there are places where these things appear the same colour. And it's only if you like the context around them that allows you to tell which is which. The root material density also varies along root branches. Okay? X-rays measure density, so in the image, the grey level, the colour of the roots changes as it moves, as you move down the branch. Probably because the material, older roots have a different response to the X-rays and so on. Okay? It's a 3D problem. So here we have volumetric images. The root and soil sample is placed in an X-ray machine, scanned, and we get a three-dimensional array of data out, a large one, something like 2000 by 2000 by 2000 voxels, yeah, voxels being 3D pixels. Yeah. So we've got a big, messy 3D data set, and in it we've got very, very small roots that are quite thin. Okay? Sometimes only 3 or 4 voxels across. Okay? So big data, small thing. How do we find this? We had produced a way of doing this previously based on tracking using level sets, I think called RootTrack some time ago. So we have something that can generate some training data for us. But again, RootTrack needed some interaction. Yeah? It was mostly automatic, but you needed to tune it, and that took a little bit of skill. So the core problem is this. On the left, you see the ground truth. This is what we got from one of our stacks using RootTrack, which we're quite happy with. If we take a neural network approach so that we can get a fully automatic solution, how big do we make the piece of image that we put into the neural network? We can't possibly put 2000 by 2000 by 2000 voxels into a neural network. There simply isn't room in the computer. So we have to do this a piece at a time. If we take a small volume and process that, something say that is 16 by 16 by 16 voxels, we are seeing the fine detail of the data. Okay? So we are able to pull out the root structure. We train a network up to distinguish roots from soil. This is the output we get. You can see lots and lots of fine roots in there, but also because it's a very small piece and it doesn't have the context to tell it that this isn't a root, because if you move away from this point, you don't see a root structure. It's not attached to anything. It also generates lots of noise. If we try to capture that context by having a larger volume, 128 cubed, yeah, then we see the opposite effect. Yeah? We don't have the noise, because when you take a broader view, it's obvious that all the little speckles are not roots. They're not connected to anything. Okay, but because we have a bigger view, we lose detail. Yeah? We can't see the small things in it because we're not focused in on them. Okay? So what we developed was a novel root detection architecture. Yeah? A new type of CNN, which has 2 branches. And I won't go into this in too much detail. The paper's out there if you want to have a look at it. It takes an input volume, right? A small chunk of the X-ray. The piece is 128 by 128 cubed, and we take a 16 by 16 by 16 cube in the center of it, the little yellow piece. Okay? The yellow part goes through at full resolution, yeah, through a network that learns to segment roots from soil. Yeah. The full 128 volume goes through a low resolution branch, which has a very, very similar architecture, and also learns to tell roots from soil. It does this by shrinking down the 128 to 16 by 16, so it loses resolution. Yeah? It's a larger piece of the image, but it's been shrunk down, so it's lower resolution. So you have a very high resolution central part, you have a low resolution but larger piece that captures information around it. And we train networks to do those two tasks, and then at the end we combine the output. Okay? Big thing about these things is everything is learned. So when I say we combine the output, what I mean is the network learns to combine the output. Okay? We give it input images. Initially we gave it data from root track to learn on, and trained it up, and it learned the weights to use in the neural net in both of the branches and how to combine them. So this is the output that we get. You can see in the small pictures of what you get from the individual pieces, the small and the large pieces of image if they're processed separately. Okay. In the middle is the ground truth, that's what we put in, which we were trying to get it to learn. On the right is the multi-resolution output. So this is pretty good. It's somewhere between the two fine detail and larger receptive field outputs. We've got most of the network. We've still got some noise, but nowhere near as much as we had in the fine detail one. So how do we fix this? Well, the network is obviously broadly right. It's doing the right thing. What we need to do is think a little bit more about how we train it. So what we did was use a technique known as hard negative mining. And what this basically means is you isolate the places where the network has gone wrong, and you provide more training in those areas. So you basically go back to the network and say, you got it wrong here, this is what that should have been. Run the training thing again, the network adjusts, and learns how to do the test better. It doesn't filter out just the things that you point at, right? It learns to do the whole task better. Okay? So it's a generic training mechanism. And at the end we got some pretty nice results from it. Okay? So novel network architecture, sensible use of training strategies. We could also do transfer learning with this. Okay? You can do transfer learning with anything. The images I've shown you so far came from what we call the small or the medium scanner. We have 3 in the Hounsfield facility at Nottingham that work at different levels of resolution. Okay? Different characteristics. We also have a very, very large scanner which will do metre-long soil columns. On the right there is an image of a taken from the large scanner. The difficulty we have with the large scanner is that the image quality is very, very different from on the small and medium ones. So the root track mechanism doesn't work so well on it. It finds it very hard to track roots through this much, much bigger and more complicated data. So if we use the large scale data, we have to use a lot of interaction, which is a problem and takes time. Transfer learning, though, means that with a very few examples created interactively of large scanner data, we can transfer what the network has learned from the small and medium scanner data and root track so that we get the results that we see on the right. So context is a problem that comes up quite a lot in the images we work with. We tend to have very large images of complicated objects and have this problem that you can't put the whole image into a neural network at the same time because there simply isn't room in the machine to do it. Okay? So this is another example of this. This is a laser ablation tomography image. The sample, in this case a root, is pushed into a laser which cuts away the end of the root. Color camera takes an image of it so that you see the anatomy of the root as it's pushed through. You see a couple of examples here. This is a complicated image. What we want to do is we want to classify each of these cells depending on their biological type. Okay? But if you take them in isolation, those cells look pretty similar. You see 2 examples pulled out of the image on the left. They're different types of cells, but the 2 images look very, very similar. What distinguishes them is their position relative to each other and other things in the image. So context really is key here. It's kind of like the X-ray thing, but a slightly different variation of it. Okay? If we process each of these rectangles, these pieces of images in isolation, then we can train a network to do the semantic segmentation thing that I showed you with the road and with RootNav, but we get some errors. And if you look at this image on the left, you can see lots of places where there are cells labelled with one colour dotted in among lots of cells labelled with another. Okay? So it's clearly where the classification has gone wrong. These the pink ones in the middle here should clearly be pink. Should clearly be blue, sorry. Yeah? And there are some pink ones here that have encroached onto the green area. I won't try and use the biological names because I'll only mess them up. So how do we solve this? If we could put the whole image into a network in one go, it would be fine, but we can't. If we split it into regions and do it a region, then we lose the context. So what we came up with was a network which did the tiling, split the image into squares, as you can see on the left, processed each one separately, and hopefully you recognize the structure of the encoder-decoder network, but then we added context sharing layers. What these do is they take data from tiles that are close to each other, and they bring it together, process it, and then put the data back. So the effect is that information is propagated across the image from one tile to another. Okay? And the network effectively learns that a particular classification is expected to be in the center, yeah? And another classification is expected to be at the side. Okay? And we produce, again, an output, a segmentation output that you see on the right hand side. Okay? So this is our larger image of our output. You have the input image on the left, you have the results in the center that we got, and if you look this time you don't see so many misclassifications. Okay? It's fairly obvious that the different parts of the root are separated from each other pretty well, certainly better than they were before. On the right is the ground truth. So this is data that again was created by a human annotator, a biologist, using one of our earlier tools, this time something called CellSet. Okay? And so we have learned to segment these images up in a way that's consistent with the human interpretation. Key thing with this is this uses 27% of the memory that it needs to put the whole image in. So this is a practical solution. So there's a number of things going on here. Yeah? We're developing convolutional neural net architectures, yeah, for a variety of tasks. Once we've done these image analysis tasks, we can extract traits from them. We can count the number of cells of different types in the laser ablation tomography images. We can pull out 3D and 2D structural descriptions of roots from the blue paper and the X-ray images. Okay? So this is the core of the plant phenotyping stuff that we do. Yeah? Using neural nets allows us to get automatic solutions to all these problems which we never had before. Yeah? What the neural nets do is reformulate the problem so that we have to decide how to learn the task rather than how to actually do it. So it makes us work, if you like, a step away from actually coding the system up, which in some senses is more appropriate. Yeah? We kind of know how to do it, we know how to give, we know what we want the input and output to be, but it's really hard to hard code the solutions. Yeah? We did okay, but you can do it better by learning it from a set of data. You can just find, sometimes, existing neural net architectures that fit your problem and train them up with your data and run them, and that's fine. This doesn't work all the time though, and quite often you hit problems like large images and small objects and context, okay, that we have here, and you need to make it practical and to improve the performance. You need new architectures and training regimes. One of the skills in this is at what point do you stop messing with the architecture and start thinking about how to train it, because both of these things can help. One of the things that's been a big help to us has been the set of first generation interactive tools we produce, because we use those to provide the training data. So that's all I have to say. I've been giving the talk. A load of people at Nottingham were involved in this, particularly Andy French and Mike Pound, but as well Razor did the X-ray work, Rabail did the work on RootNav2, okay? And we have our biologist colleagues Craig, Darren, Marcus and John. Yeah? Ezzy Bunsen developed the LAT system. Thank you very much.
Thank you, Tony. It was funny, one of the first slides was minimal human interaction. And this conference, one of the nice things is the human interaction, I think. That's also why we like to shift the program a little bit and continue with Shawn's talk, and after that with David's talk. And then we end this afternoon session before the coffee break with another recorded talk of DJ Stricker. So a little bit turnaround to have the human interaction now. Shawn from Spain, he will talk about the MaizeScanner that he's been working with. Looking forward to your talk, Shawn. And here's the microphone.
Thank you. Thank you very much. All right. So I want to take a pause for a second and just focus on one of the phrases at the end of Tony's talk, which I thought was really good. He said that this was a practical solution in one of his things. So sort of my task, I was going to present about the MaizeScanner, then I sort of expanded a little bit more. So I want to talk about some practical solutions to phenotyping, and it's sort of in the context not of advancing science, but of supporting a network of collaborators that are trying to advance their science. But anyways, it's a slightly different perspective. So introductions. My name is Shawn. I'm at the University of Barcelona. I'm from the United States, from Texas originally. And my PhD was in ecology, focused on remote sensing, in a laboratory, the Center for Spatial Technologies and Remote Sensing, that was very technically focused. And I and my PhD advisor were the only people interested in plants. And now I'm in the, not ecology, but the plant world. And I'm trying to support all of the technology around this and try to advance that in different ways. So I want to jump through scales of observation and application. Quick talk about RGB. I see color images are growing and growing. I wonder if anybody will talk about multispectral data ever again as we move forward and onward and back to the basics, I suppose. And then I have different examples of applications and software and things that you can find here and all these different links I threw up. So my aim joining the phenotyping community was to improve data quality and data quantity. So we had at first a bottleneck in collecting data. Then we have different bottlenecks in processing the data, and still there's some bottlenecks in data science, but I think we're getting to the point where we need to integrate the data a little bit better with smarter processing and more efficient process systems. So looking at proximal-based imaging, high-resolution RGB, mobile platforms, mobile phones, or other things that move, UAV RGB imaging, and then what can we do on the backside of that to improve data collection for better consistency and efficiency? So I started out looking at and throwing out different ideas at the leaf scale, the ground scale, and the aerial scale. You can see here some maize leaves, some wheat fields, and drought and irrigated. I'm going to focus mostly on ground scale and aerial scale, though the MaizeScanner initially included some tools for leaf level assessments of disease impacts. So stepping back, looking at RGB images, this comes from a casual dinner conversation I had with some researchers at a networking conference years ago. And it was Anatoly Gidelson who was saying, well, actually, they have really good calibration, good color calibration, and they cover perfectly photosynthetically active radiation at very high resolution. So there's tons of information there. It's a 3-band multispectral sensor that covers one of the regions that we're most interested in. And of course, you can make an argument for hanging a hyperspectral SWIR sensor off of your drone, depending on what you want to measure, but not everybody can afford that. So this was the first concept for our MaizeScanner. It was a collaboration with CIMMYT. And there was these general tools that we put in that we thought maybe would be useful for people. People had asked us for access to this really old set of codes developed by Casa de Sus called BREPIX and some extensions of them by some of our collaborators in Zimbabwe, the crop senescence index. So we put together these general tools. The same MaizeScanner also included this whole other section that quantified the level of infestation or impact of maize lethal necrosis in maize plants in Kenya based on trained data, ensemble, machine learning, Nobody ever used that. But the software and the basic tools that it provided in a user-friendly format has been downloaded several hundred times. And I get questions about it almost every week. So apparently the basic uses of quantification of RGB indexes was something that a lot of people wanted, and our collaborators weren't entirely convinced that RGB images could quantify disease impacts. But anyways, so that hit sort of a sweet spot. This is an example of the crop senescent index or maturity assessment, which worked really well for quantifying different moments of senescence. Again, a little bit backwards, so higher values are senescent. So this was the idea for moving forward and working closer with the phenotyping community, with some private companies, and developed something that were things that we're measuring in the field. They had doubts about how consistent they were in the field, and it definitely took them longer than they wanted. So we made the serial scanner, so RGB-based improved models for specific trait assessments that they were interested in. So they wanted a tool for optimizing early vigor, for counting wheat ears, and for maturity and crop senescence assessments. And so we worked with them on developing an algorithm that would work for each one. And they said this should work on a standard field laptop that's not too expensive. So we had to adjust some of these and provide mechanisms. And we even did a video publication. I don't know if anybody here has ever done a JOVE. We did a Journal of Visualized Experiments video publication, which was lots of fun, and I'm convinced that nobody's ever watched. But anyways, check it out. It's really nice. And then we started throwing around some ideas with our collaborative networks in Africa, many of which had a mobile phone but didn't have access to a computer, or they didn't have a laptop, but they had a computer they could use in the laboratory. So we started looking into how to explore the uses of Open Data Kit for collaborative networks of precision agriculture and phenotyping projects. So we threw out this idea. So if you're not familiar with it, ODK Collect is an open source Android software. You can make a form, you can take images, RGB images or modified RGB images with the data. It goes from there into an aggregate server, which ODK can help you set up. You can publish it to Google Fusion Tables, or I think it's Google Sheets now. You can process that data and update it. Here's an example of a fisheye lens attachment that we got for a mobile phone for doing leaf area index assessments based on CanEye, which was developed here. And then you can publish that automatically back to look at heat maps, so visualizations of your data in real time. You can see in real time visualizations of the data collected by other people. So if you're looking at a pest spreading across, in this case it was Zimbabwe, you can collect pest assessment. You can publish that, and you can see what your neighbors are saying, how much they got, or how much other people in other parts of the country are experiencing. So we tried to adapt some of our tools to this, and we thought it was a great idea. So the BreedPix Fusion and Serial Fusion are the exact same versions of the software that I showed earlier, but this would be an example where you go to the field, where before you go to the field, you turn your computer on and you launch the software. The software will look for the data on the web, at the website that you're going to publish the data from the ODK aggregate. And as you submit data, it will download it, process it, and upload the results. So on your mobile phone, you can take a picture of something, you can upload it to the website using the ODK form. It'll automatically process the data and then pin the results there. So you can have, let's say you're PhD students out in the field all collecting data, and you could sit there in the shade and you could see them collecting the data and the results of that analysis as they measure wheat ear counts per square meter in the field. So again, this is something that we thought would be great. We shared with our collaborative networks, and I'm not convinced that anybody used it, but it could be potentially useful in a different scenario as a test of a concept. So this is an example of collecting the data, and so this would be running on your server. So we called it sort of cloud computing light, because you're running the cloud-based processing on your own computer. Collecting the data, this is doing nadir looking downward assessment of fractional vegetation cover, and then it's uploading automatically the results in a different website where you can view it in real time. So we took this to the next level in a collaboration with the FAO, looking at changes in leaf area index in relation to the fall armyworm pest infestation that was crossing Africa. The original concept was to look at multi-scale assessments of leaf area index at a continental scale using top-down and bottom-up approaches. So Sentinel, Planetscope, different drones, multispectral sensors, and then a fisheye adapter on a mobile phone running the Open Data Kit collect system to collect data, send it back so it could be processed, and then share it back in real time. In the end, there was lots of clouds in Africa, so we have spotty satellite coverage, affecting even daily NDVI values from Planetscope. 2 out of 3 of the countries changed their drone laws after the project started. We put our multispectral sensors on a 5-meter pole and ran through the fields, but the fisheye lens adapter collected really good data. So we have some calibration and validation of this working at different levels, and hopefully that'll be coming out soon. We did a poster recently, I think. And so as we collected more and more data with the Open Data Kit, we started having a bottleneck in our data processing, which was actually downloading all of the data, reorganizing it into different folders in order to batch process it using a separate standalone software. This was the case with the CANI software. We didn't manage to connect it into our Java system because it was already its own software. So we just download the data, reorganize it. So we developed this easy ODK downloader, which is publicly available here. So if you publish a data set using the Open Data Kit, you can attach labels to it, and this will simply download the data to scrapping software, rename it based on different formats, and then put it in different folder structure names. So this was saving us lots of time. Moving forward to our next collaborative project, which wasn't in sub-Saharan Africa, but across northern Africa. The example here, there we go. So we were looking at foliar disease assessments in greenhouse crops across northern Africa. And in this case, they didn't want to quantify a disease impact for phenotyping purposes. They wanted to identify the disease from a selection of 15 or so diseases that were very common, identify it, and then provide as user feedback immediately the results of the disease and how best to treat it in their region. So this is an ongoing project, and we needed to validate the data. We also needed to collect massive amounts of extra data for developing a deep learning convolutional neural network sort of system. So, the ODK downloader will allow us to receive images collected by 5 different countries continuously over during the pandemic over multiple years, bring them together. We can quality assess them, label them. We had them in the ODK take 1 picture of the whole plant for visual assessment, and then 5 pictures of separate leaves on a specific background. So very easily we can have a plant pathologist sit down and validate the quality of the input data before we train these models. And so the idea is that this will collect a database that doesn't exist of thousands and thousands of pictures. I think we're up to maybe 30 or 40,000 pictures. And then hopefully going forward, that'll allow us to develop these artificial intelligence models. I wanted to throw up a quick note about UAV phenotyping and some of the other tools that we developed related to high throughput phenotyping products. So I was telling some people about different 3D systems using structure from motion. I'm glad Bernie included his comparison of LiDAR and structure from motion. You know, I'm sort of on the fence, like do I need LiDAR or is my 10 million point cloud from structure from motion going to be good enough? Because what are the benefits of them? What are the differences? I'm curious about the differences in penetration, specifically if you look at cereal crops versus forests. Does it make sense for forests but not cereal crops? We can talk about that later. So how to batch process things and how to get through it quickly so that you can get on to really the interesting part of the science, because we're still collecting a lot of the same basic data across all of our different platforms. So we developed the Mosaic tool. Now this was initially launched, I think 8 years ago, when an RTK GPS was 15,000 euros that we didn't have. Now I think you can pick up one for 2,000 or 3,000, so maybe it's obsolete. So this is non-GPS based phenotyping. And it basically just draws out a segmented plot of your entire phenotyping network. Then it goes in an automatic mode, in semi-automatic mode. Then it allows you to manually adjust those as needed. And then we have built-in tools for different sensors to automatically process the microplots or save them as a useful database. And so this took us from 9 months of post-processing to about 6 weeks for maybe the 20,000 plot images that we have to process a year. So this is also available if anybody wants to test it out. Again, people try our software in ways we never intended it, and sometimes it makes it better, and sometimes it just makes it crash. So feel free to reach out. We've also been working with local forestry services. Again, they asked me to sort of batch up together some code that we had written as part of a project to make it useful for them so their technicians could process these standard sets. And so this was just some assessments based on a specific drone that they wanted to use. So an RGB drone and an agrocam modified NDVI camera to calculate a couple of different vegetation indices from RGB, from color infrared, from a digital elevation model, extract out sort of canopy profiles for looking at pruning techniques and some other things that they wanted to. So not too difficult to package it into a little plugin for them, and that makes this a little bit more operational than giving them slabs of code that are still intimidating to some people, I think. So again, this is the collaborative research of a bunch of different people. I'm part of the Integrative Crop Ecophysiology Group at the University of Barcelona, led by José Luis Arauz. We have a good international team of researchers there. We are also part of the Agrotecnio Research Center based in Lleida and the Institute of Water Research at the University of Barcelona. And that's all.
Thanks a lot, Shawn. Any questions for Shawn? Shawn is still walking in the field as a cowboy, but
it's hot in Spain. Somebody was asking me.
He does that already for many years. So, are there any questions in the back all the way? Yeah.
Just a very small question. It's the data that you are collecting. It's specifically for research purpose. I can imagine you cannot download it for commercial or other purposes.
Well, in all of these, so this is sort of a project-based rather than a science-based presentation. So in all these projects, these international collaborations and stuff, we're required to make freely available and open source all of our code and all of our software. So this was supporting our collaborative networks and seeing what we could do for them. People do ask about commercializing it. There are commercial companies that do these sorts of things that happen to be sponsoring this workshop, and I'm sure their data products are more advanced, but you might have to pay for it.
We may have time for one more question. I may ask David to come here already. But he's there. Yeah. Okay. Because if not, I propose we continue. Thanks a lot, Shawn.
Thank you.
Talk.
Thank you.
Shawn will be still here, and we will have time for coffee break. David will be maybe the part why this is a sanitary session, which was an interesting term for me. Sanitary traits. But I don't know if that's really related to you, David. But yes. But David will talk about spectro-imaging and compressed learning. So in a 20 minutes compression, looking forward to your talk.
Okay, thank you. First of all, I thank all the organizers for this invitation. That's super nice to be here again in Avignon. I think 14 years after my first visit, when I was starting in this domain. I'm heading the Imorphen bioimaging group. This is in Angers, and we are working inside the Phenom network and in cooperation with INRA, of course. Today, rather than let's say a general broadband talk, I will put a focus on a rather low TRL new sensor called Spectro-imaging. And this was done in the framework of the PhD of Clément Douard, who is now a research engineer in CEA Grenoble. It was in collaboration with Carbon B Company and Université Lyon 2 University. Okay, so we all know detecting disease is a complex task, which sometimes completely is out of scope for the human eye. And this is why most of the sensors which are presented in these days include several modalities, classical ones like we've seen before, but also new ones like the spectro-imaging system that I'm going to give you focus on today. Spectro-imaging takes benefit of the three-dimensional information that are present in a spectral scene, XY 2D plane, plus spectral slices. And we have here, in some of the hyperspectral imaging, a very high resolution in the spectral domain. There are several sensors for doing this. Push broom acquire spectral image slice by slice in this way. And a drawback of this is that it's very slow acquisition. It requires static scenes. And somehow there are advantages on snapshot imaging, which are capable of acquiring cube in a single acquisition. This is exactly what CETIS, computed tomography imaging spectrometer, is doing. It's based on diffraction grating, spreading out the different spectral layers onto the sensors in this way. A CETIS image looks this way, where you have at the center a kind of classical zero-order image, and you have diffraction orders, which look like a spectrum in the different directions. So you may wonder, where is the spectral cube here? Actually, it's necessary to reconstruct it. So you do the acquisition, you get the CETIS image, and then you need a reconstruction based on some specific algorithm called expectation maximization algorithm, rather specific. And also, especially time-consuming and non-perfect, meaning that they can do errors when they reconstruct the image. This CETIS has been introduced, let's say, some 30 years ago and has been applied in different domains of spectrometry. And what was striking for us is that it was even used when only a classification task was targeted. And when only a classification task is targeted, maybe we don't need to do the reconstruction. We can directly use the CETIS plan. So instead of doing machine learning after a reconstruction of the hyperspectral cube, can we directly use the raw CETIS image? So that's the question we addressed in this work, to save some time and some memory load, not to do the reconstruction, which is only here for human eyes. As a use case, we consider apple scab, which is a fungi, which has high contrast more in the infrared and a bit in the green domain. As a disclaimer, I want to say that we are at, as I said, low TRL here. It's a first use in compressed learning and in application in plant sciences. So we do simulation, and for now, lab application, and we start to move to field canopies, as you can see here on the right. So we need to create the data set, which we do this way. We take healthy images. We have an hyperspectral cube simulator, and then a CETIS simulator. So everything is simulated physically. We take tons of data set of healthy leaves, and then we simulate some scabs this way to generate scabbed images in this way. So since the lesions have an ovoid shape, we generate some funny stuff in the Fourier domain to mimic this kind of lesion and to produce some automatically annotated images in this way. The spectral slides are done based on real images acquired on a push broom hyperspectral imager and on real leaves. And we record, after manual segmentation, the healthy spectrum, average spectrum, and the scabbed spectrum. In the end, we create 3,000 healthy leaves. reconstructing this realistic spectrum in the visible and infrared domain. And to add a bit more of realism, we simulated a kind of severity of the amount of fungi that we have between an healthy one and a scabbed one, which is kind of realistic because the fungi acts like an optical filtrum somehow. Concerning the CETIS simulator itself, basically we mimic the way the diffracting grating is happening this way. So we generate each wavelength this way. I don't know if I can play the small video. Can I have some help on this? Maybe it's not playing. Yeah, exactly. Maybe you don't see it. Now it starts to appear. So it was mimicking all the projections, like if you had card and they were spreading out on the. So then you have the final simulated CETIS. Okay, so now we have our data set with healthy and scabbed. And we have a binary classification in the end where that we reproduce on several severity level of the disease. That we come to the machine learning part where we'll do supervised machine learning, similarly to the talk of Tony previously in the afternoon session. We feed with healthy, unhealthy, healthy, unhealthy, and after the algorithm is trained, we want to predict some unknown label on these images. We use neural networks as believers that it can be useful for hyperspectral imaging and for disease detection, especially for signals which are invisible to human eye and ear. We considered a very classical convolutional neural network architecture here to start with. That's a VGG, that's a classical convolutional neural network. And to have a baseline of comparison, we start to apply this network on the reconstructed hypercube. So let's have a look at the performance. So this is the image, the real slice, the reconstructed slice, and these are the spectrum that you can have after reconstruction. So you can see we lose a bit of detail either spatially and spectrally, but however, it's rather good. And then if we feed our CETIS image, so we got a little bit less performance, a bit less here, of course, but you have to remember that we saved the reconstruction. So here, the black line takes a lot of time, while actually it's only 10 milliseconds per image for the VGG based on the CETIS image. We can do much better than this if we put a little bit of intelligence and see where to look. Basically here, the network was only looking at the order 0, the center of the image, while actually there are separate information in the center of the image and in the spectral part, which is in the order 1. And in this case, actually, if we train only on order 0 or order 1, the convergence, the speed of convergence of the algorithm is clearly different. So it only focuses on the fastest answer, which was here order 0. So to take benefit of the two spatial and spectral information, we proposed a CETISNet, which is separating the different spatial and spectral information and then lately fusing them. Because we know that convolutional neural networks are invariant to translation somehow, we align the projection to avoid that the network looks for information in here. And we consider that we have spreading the spectrum, so we deal with non-square kernels in this case. Okay, so we put a bit of thinking of how we design a new architecture as it was promoted in the first talk of this afternoon by Tony. And these are the results that we obtained. So we reach almost the performance of the reconstructed field, but remember, here this is obtained 100 times faster than if we had done the reconstruction. Okay, so this is published in this paper where there is no Fiji application, but all the codes are available on the dataset and the simulator. So if you want to play with CETIS, you can go in the paper and look at this. What we can remember about this is as compressed sensing is shown for the first time here with CETIS. We propose a specific architecture mixing spectral and spatial information, and we are on our way for a transfer from simulated to real ones. We have some real data set where we have trained on synthetic data and apply trained on synthetic and applied on real one successfully in one of the 3 papers cited here. As a perspective, so if you are excited by CETIS geometry, actually there's a lot of funky stuff you can still do because there's many other gratings, optical gratings that you can think about. I'm looking at Antoine Fournier from Photonics Bretagne. So many different geometry here that you can think of and which may enhance the spectral information. And my more general comment is that I believe that compressive sensing has offers a lot of opportunities to develop new sensors in agrophotonics, to relax constraints on the instrumentation, and to reduce the data load, the CO2 footprint, and with very limited loss of performance, as I demonstrated today, only one example on this low-cost hyperspectral imaging, because it comes for the cost of a simple Raspberry. Okay. So this is my talk here, where we provide a lot of tutorial on what we do in the group, so you can go on the YouTube channel and follow us. And this is something more for French-speaking people.
Thanks a lot, David. Deep human attention. Any deep human attention? Any questions attentive to David in a compressed talk? Could start off with a simple one, but what was your source slide that you were using for this?
Yeah, here it was classical plasma light for indoor lighting. But when we go in the greenhouse for what we did for the canopy, we move to natural light, which indeed needs a bit of more training because we can have kind of variation, of course, during the day. So this would, if we move from indoor to outdoor, this will be what needs to be, let's say, further data augmented to compensate for global variation of the spectrum, which hopefully will still keep some contrast in what makes the difference from the SCAB and LFI1. Thank you.
Any, Bernie?
No, very interesting, and I'm quite excited to see the progress in this technology. I wanted to ask you about the bottleneck in time for the calculations and the difference between the compressed and uncompressed methods. Is that on the reconstruction of the diffraction image, or is it on the way that the neural network deals with the data?
Yeah, the neural network is here only doing the inference, so it can be very fast. Actually, it could even run on a Raspberry itself, okay? But the expectation maximization algorithm, the reconstruction, which is an iterative algorithm, is the longest overload. So that's why if we can learn directly in the raw data space for classification purposes, that's a gain of time, which is interesting.
In the system, did you lose in terms of spatial resolution because you project everything on the same matrix?
Yeah. Thank you. Here I only put the focus on using the CETIS itself, but as you have seen on one of the early slides, there was actually infrared itself, RGB, plus this channel. So we believe it's interesting to keep this free Raspberry cam and fuse the information, not to lose too much spatial information, because obviously when we only use the order 0, it's very poor in terms of spatial information.
Yeah, in the back.
Thank you for the presentation. I have a question about the task of machine learning. The task was just a simple classification between healthy and unhealthy leaves. So I think it's a simple task. I have a question if you are wondering to know if using this kind of data set will do some good job in segmentation, because it gives some information from other hyperspectral channel, so maybe it gives more information on the segmentation. So if there is some work done on segmentation, not classification. And I want to know if the purpose of this job just to save time and memory, or because I see that the task was simple, classification is really simple, and to gain time and memory, it's not really a purpose in that one.
Okay, thank you. I start by the second question. If you have already played with some hyperspectral imaging with the push broom stuff, you know you will lose a lot of time to do this simple task, because it's 1 gigabyte by image to be calibrated, to be transferred to some data processing stuff. So the gain of time is not something. The fun is to say, what can we do with this kind of representation which dates back from the 90s, which has been left apart, and are our application in agrophotonics interesting for this stuff? That was the starting point. And here again, if you had to do this with a push broom hyperspectral camera, it will be a hell of a job. Yeah? But I agree with you that it's this compressed learning works because we do a very simple classification task. If we were moving to a segmentation here, the spatial resolution in the cities, that was one of the questions also Fred earlier, is a bit too tight. I believe that combination of the 3 channels of our camera is something nice. So we could have a kind of raw detection of disease, non-disease, and then call for the other camera to look for where they would be, especially in the infrared, which is, as we know, the best contrast in this case. Thank you.
There may be time for one more question, because David was nicely in time.
I was very compressed.
Yes. If not, thanks a lot, David.
Thank you.
He's also still. And as announced, we will have a nice coffee break, because the next talk is actually, instead of DJ, we have a talk by Xiaoyu Zhi, who will talk via a pre-recorded session about estimating photosynthesis attributes in sorghum. And he also uses a hyperspectral sensor. Yeah, if you would like to have a longer coffee break, you could also, from my part, join this talk, because it's interesting, but also there is some room to be quietly move to a coffee session on time. But okay, half an hour, 20 minutes. Xiaoyu Zhi, looking forward to your talk.
Hi everyone, this is Xiaoyu Zhi. I just graduated from the University of Queensland in Australia last month, and today I'm going to talk about one of the chapters in my PhD thesis, estimating photosynthetic attributes from a high-throughput canopy hyperspectral sensing in sorghum. There are 3 sections in my presentation. And the first one, I'm going to introduce a little bit of the background for this piece of work. As we know, increasing yield is still one of the most important topics in agriculture to make sure the food safety, because of the increasing demand of food globally. And as we know, yield can be broken down into accumulated radiation uptake, radiation use efficiency, and harvest index. As we also know that in cereals, harvest index has nearly been maximized since the first Green Revolution. And if only increasing accumulated radiation uptake can cause trade-offs, so researchers has recognized the increasing radiation use efficiency is an important avenue for improved yield further. However, to improve radiation use efficiency in breeding, we need to understand its genetic control. Well, to understand its genetic control, we need a high-throughput phenotyping tool to determine the efficiency of energy conversion through photosynthesis into biomass. Photosynthesis is determined by light interception, water, and CO2 assimilation. Light interception is influenced by plant architecture, such as leaf area, height, and leaf angle, and transpiration is influenced by stomatoconductance, at the leaf level, while at the plant level, transpiration area is also one of the factors for CO2 assimilation. It's mainly determined by biochemical limitations and physiological properties. In my thesis, I was focused on to modify leaf angle for improving light interception, modifying leaf size to improve transpiration efficiency, and also biochemical and physiological properties for improving CO2 assimilation. But today, I'm not going into details of the first two components. I'm going to talk about the biochemical and physiological properties. Well, to improve biochemical and physiological properties in large breeding programs, we need high-throughput to obtain this data quickly, which is still the bottleneck for this part, how to obtain these biochemical and physiological limitations quickly. So in C4 photosynthesis pathway, there are coordinated limitations in mesophyll cells and also bundle sheath cells. And in bundle sheath, photosynthesis rate was limited by maximum Robisco activity, which is Vcmax. And in mesophyll, it's limited by maximum PEP activity, which is Vpmax. And the regeneration of Vcmax and Vpmax are limited by the maximum electron transport rate, which is Gmax. However, in the practice, if we want to obtain these three most important photosynthetic biochemical limitations, one sample will take about 1.5 hours. It's impossible to obtain all of these traits in hundreds of breeding lines. Fortunately, hyperspectral reference is highly associated with plant biochemical and physiological features. Therefore, objectives of this study were to estimate biochemical limitations, including Vcmax, Vpmax, and Gmax, and also physiological features that are highly associated with the photosynthetic capacity, including specific leaf nitrogen, SLN, and leaf mass per leaf area, LMA, by using the proximal hyperspectral canopy reflectance, which was captured by the spectrometer attached to a mobile phenotyping platform. To do that, two field trials were conducted to collect ground truth measurements and also the proximal hyperspectral sensing data. Well, to predict Vcmax, Vpmax, and Gmax from hyperspectral sensing data is not straightforward. We need a C4 photosynthesis model to fit the three photosynthetic parameters first, and then use a regression method to predict these traits from sensing data. So I used a C4 photosynthesis model by combining ACI and AI curves to fit these three parameters. And just a short introduce of this procedure, that ACI curve is the CO2 response curve, which is at the right-hand side, and the AI curve is a light response curve at the left-hand side. From the AI curve, we can see to Gmax, which informs the electron transport limitation. And as we can see that the asymptote can be best fitted by the electron transport rate from AI curve, and the initial slope can give us a Vcmax and Vpmax. So by combining ACI and AI curves, we can fit these three photosynthetic parameters. While we have the ground truth data and the sensing data. And two approaches were used to link these two types of data together. And the first approach was used vegetation disease-based stepwise multilinear regression. And the second approach was reflectance-based partial least squares regression. Well, the first result is that is a variation of a photosynthetic capacity among the genotypes that in the two training sets. So from the box plots, we can see that there was wide ranges of VC max, VP max, and J max in the sorghum diversity panel, and so as SLN and LMA, which potentially give us great opportunity to use sensing data to predict these photosynthesis-related traits. So secondly, can we predict variation in photosynthetic capacity using the sensing data? Well, by using the stepwise multilinear regression, it always suffers from the collinearities between the predictors. So there are 16 vegetation indices that I calculated from based on the information from literatures that showed they are associated with photosynthesis. Well, we can see there are a lot of strong correlations between these variables, such as these three are highly correlated with each other. So it would be problematic if we use the whole set of vegetation indices for the stepwise multilinear regression. So to avoid the collinearities between these variables, PCA was conducted before stepwise multilinear regression. So a subset of vegetation indices were selected to go into the stepwise multilinear regression. And the regression result shows that all of the models for the traits of interest are significant, although R square was not very high. However, root mean square error was acceptable, except for VP max. Well, further studies would be further studies are needed for this parameter based on this approach. And then by looking at the predictors for each trait, red edge was highlighted in the predictors captured by the best models for predicting traits of interest, suggesting its high association with photosynthesis in sorghum. And also another vegetation index, PRI, was reported to be associated with light use efficiency, was one of the predictors for VC max. And also the reflectance at 760 was shown to be associated with chlorophyll fluorescence, was captured by the best models for VP max and J max. So these parameters can be targeted in the future studies for predicting photosynthesis by the hyperspectral sensing data. Well, by using the second approach, partial least squares regression, models for VC max, VP max, and J max are improved based on R square values and the root mean square error. And also looking at the loadings of the different wavelengths for the models of these three photosynthetic parameters, the red edge region was also highlighted, which is consistent with the result by multilinear regression. So same as the result for the 3 photosynthetic parameters, the models for SLN and LMA were also largely improved by using partial least square regressions. And also the red edge region was also highlighted by partial least square regression, which is also consistent with the result from the multilinear regression, and suggesting that in further studies, the red edge region can be a good contributor for predicting photosynthesis. So thirdly, can the partial least square regression across the training sets extrapolate to GWAS trials? So by using partial least square regression models, I predicted the trees of interest into GWAS trials, and it showed that the predictions for the trees of interest are in reasonable and comparable regions with high heritability. And there is a very interesting result. The heritability for the trees in GAT2 were consistently lower than those in GAT1, which is expected because genotypes in a trial of GAT2 were mostly hybrids, and hybrids usually have lower heritability than inbreeds. So in this way, somehow the result validates the reliability of this method for application in breeding programs. And the last result, since I had the GWAS trials and I used the predictions for GWAS analysis and identified potential candidate genes for VCmax and Jmax, because these two parameters are the most important photosynthetic parameters in sorghum. And then the result shows that the candidate genes for Jmax are indeed associated with electron transport. So this is a very good validation for this methodology. And conclusions for my study: hyperspectral sensing measurements can be used efficiently to capture the photosynthetic parameters in large breeding programs. And by extrapolating this method to GWAS trials, I found the predicted trees showed medium to high heritability, and GWAS using the predicted values identified 4 QTL for VCmax and 2 QTL for Gmax, and candidate genes within 200 kb of the VCmax QTL were involved in nitrogen storage, which is closely associated with the rubisco, while not directly associated with the rubisco activity per se. And Gmax QTL were enriched for candidate genes involved in electron transport. So in summary, this study, the method can be used to accelerate screening the varieties that have a high photosynthetic capacity in breeding. And in the end, I'd like to acknowledge my advisors and colleagues during my PhD for their help and guidance in my studies. And thanks again for having me here today. And questions are welcome.
The questions are welcome, but I understand she is not online to answer questions, so we can discuss questions in the coffee break. We continue with semantic segmentation by Benoit for crop production. Benoit, looking forward to it, and then the session is yours.
Thank you, Onno. And welcome everybody. So I will talk to you about semantic segmentation. So we had some information about this specific task with the presentation by Tony. And here we focus on the crop description. So semantic segmentation is a task that is dedicated to classify each pixel of an image in a predefined set of classes. So we can define which classes we want to define, and then each pixel of the image will be put in one class. So it's a powerful way to describe the semantic content of an image. So it can be applied to RGB images, or as we have seen earlier this afternoon, to tomography imagery. But here we will focus on RGB images, which are the most common images. So actually, it's a task that is very similar to visual analysis. So it's usually a first step before higher descriptions and processes. So we first try to identify in the image the different patches and shapes, and then from this we can extract the other traits. And for this task, deep learning is particularly powerful. So some years ago it was done using filters and colorimetric approaches, and now deep learning really took the place and is used for this task. Then, compared with other deep learning tasks, such as image classification or object detection, as we have seen with Etienne, where we want to identify objects in the image, the labeling task is very complex, because we have, when you want to train a model, you have to paint each pixel of the image and allocate it in one class. So it's very long compared to just drawing boxes around ears or objects. So it's not yes, I have nothing against Etienne, but it's more it's more difficult for the labeling task. And therefore, there are fewer data sets that are available for this kind of applications. And so usually it has also been less studied than a classification or detection task. And therefore, the models and all the parameters are usually less optimized than for other tasks. So here, the presentation has two objectives. One is to share with you some developments that we tried, so some attempts to optimize the segmentation process. And the other is to share with you some applications and some directions that we want to promote in the coming year. So one part that is more methodologic and one more applied. So first, as I said, we need to train all these deep learning models. We need data sets that will represent the ground truth, as we discussed with François, with, if possible, large and diversified data set that will be considered as the reference and from which we can learn the models. So that's something that we started since 2019 to constitute very diverse data sets of images, which have been labeled in two classes, one vegetation class and one background class. And then, saying that, we had to decide what we meant by vegetation, and we decided to say, okay, vegetation is all standing vegetations. It can be green or not, leaves, flowers, fruits, etc. As long as it's on the stem or it's above the ground, it's plant. And all the rest is considered as background, so it can be soil, crop residues, or stones, for instance. Once we have defined what is vegetation and what is background, we collected images, and especially Simon Madec collected images from different countries, from different institutions. And also each each of these partners use different sensing systems, such as smartphones, digital cameras, literal, phenomobiles, even fisheye sensors, cameras. So all are RGB, but with different focal lengths, etc. We also have different sensing configurations. Some are upwards, downwards, lateral. And last, we considered a large set of crops and vegetation types. So it can be a natural vegetation or different kind of crops. And from this, yes, we collected around 4,000 images. So it's small patches of 512 by 512 pixels. Each of them has been manually labeled in these two classes. So we used it, that's the data set we will use to assess our model, and it will be made available hopefully in 2022 for to benchmark different models. So the general framework when you want to build and then assess segmentation model is first to define your baseline and then see if you can improve its performances by optimizing some of the components of the model. And then you evaluate the improvements you can get, and then you optimize your performances. So this application will be done on this simple two classes model, and then I will show you how we can extend this simple two classes model into a more complex model. So first, as mentioned earlier this afternoon, we will not design new architecture. We will use a commonly used architecture, that is the UNet architecture, and which has the interest of considering both fine resolution and also larger context around the pixel. So it's quite commonly used for segmentation tasks. And so using this, setting this architecture, we will see how to improve different elements. So one first element, I will show the baseline model and then detail which parameters we will try to optimize. So our baseline model is evaluated with 3,000 images as a training data set, 400 for validation, and 600 for a test. So we try to choose quite standard parameters values, and we saw how the quality of the... Okay. Yes, here. Nobody pushed this button before. So yes, so we can see here as a function of the number of iteration how the quality of the training is going. So if the value is low, then the performances are better. So here we try first to train on many epochs, and then we decided to stop before at around 200. And from this we got these performances. So again, the IOU was defined earlier, and here we have a mean value of around 0.73. So that is actually quite similar to a publication done by Zurich that I discovered yesterday, and was quite it was funny to see that we have the same kind of results. So having this baseline model with standard parameters, we will try to see how we can optimize some of them to improve the performance. So the first element we can try to optimize is data augmentation. So we do not change, we do not touch the architecture, but we can play around. So one aspect is data augmentation. And for this, we wanted to test a package that is RandAugment, which provides different, a good framework to work with data augmentation. So data augmentation is, okay, we have this input data as training data, and we will change them a little bit to have more robust results. But saying that, we can have a lot of possibilities to modify our input data, and therefore we have to optimize some of the parameters of this transformation to have something that is not too far from the reality, and that could decrease the performance. So this RandAugment has two parameters, the number of transformations and the magnitude of each transformation. So for this, we try different parameters, and so we use for this Optuna, which is a package that is designed for machine learning parameters optimization, because that's a problem, a common problem with machine learning techniques. It's that we have a lot of parameters, and therefore you need quite sparse and efficient estimation optimization processes to explore all the range of possible parameters in an efficient way. So with this, we were able to improve a little bit our baseline model by choosing the best magnitude and number of transformations. So we improve a little bit. The next step was to pre-train the backbone of our model. So the backbone is, by default, was the ResNet 50, which has been trained with different. So it's a set of transformation of data with weights, but it has been pre-trained on a data set that is very generic, that is ImageNet, where you have boats and cars and ducks and etc. So very different from the kind of images that we will really have in hand, that is vegetation and only vegetation. So the idea was to pre-train this backbone on data sets that are more similar to what we will see. So we do not have ducks, but but the ducks was the example of this publication to use self-supervised pre-training as a solution to optimize the backbone. So the self-supervised is a self-learning is an interesting technique, because you just provide to the algorithm a large set of data, and of the kind of data you will have to process, and it will automatically select it will optimize the weights of your model by itself. So it's quite convenient because it's automatic. And okay, and so for this, we applied it on a data set with 30,000 images. So we collected 30,000 images on all kinds of crops that we collected in previous seasons to try this self-learning process. So in comparison, the ImageNet dataset, it's 50,000 images, so it's the same kind of volume. Okay, and what we can see is that the model performance improved, and then we had to stop because it was because the workshop was coming. But we see that we can probably improve still the performances of our backbone and therefore of our prediction and segmentation model. So we think it can be also a good way to improve these segmentation models. So here what we get, it's a very slight improvement compared with the performances of our baseline model, so we see. But we can expect that the performances will be better if we let more time, if we give more time to pre-train this model, and maybe adding also more vegetation data set, because now we have large data sets, so we can try that. So if we combine these two optimizations, so one optimization on the backbone and one on the data augmentation, we can see that we improved a little bit the overall performances of our segmentation model. So it's as often the improvement is not dramatic, but it's quite promising. And so it's sharing. And we can also try some other model parameters as published by the team of Andreas. So we will also try to play with the loss function and architecture as there are new architectures that are being released. A good point is that the Optuna package was efficient to optimize some of the parameters. So that's an important asset. Then we have to point also the limit of the quality of the images, because this segmentation of images, even manually, is very complex, because sometimes we have shades, we have part of the image, we don't know if it's really standing vegetation or if it's in the ground. So there is a limitation for complex scenes, complex images. And however, we consider that the performances are good enough for many applications that I will show you now. So the mean IOU seems not too high, but if we think about pixel accuracy, we are probably around 0.93, so 93% of precision. So in fact, here I presented a binary classification, so with only two classes, but we can apply this kind of segmentation to more complex analysis of images, of our RGB images. So from one image, we can first identify what is vegetation and what is background. So that's what I've presented. But then from vegetation, we can try to separate weeds in one hand and the crop. And from the crop, we can also identify different kind of organs. And for each organ, we can also segment the green and the senescent parts. And from the background, we can also define several classes. So it's very powerful as soon as you define specific models to segmentate each of these. So that's an example of composite image with a class with ears, one for the stems, one for the leaves, and one for the background. And from the leaves, we have a separation between green and non-green. So that's running now in an efficient way. And with different applications, so the most obvious application is the green cover fraction estimation. So we classify all the parts that are green, and so that's quite easy and efficient and accurate. Then we can work on the senescent fraction of the vegetation, or only of the leaves in this case. So we remove all the background, all the wheat heads, all the stems, and we only focus on the leaves. And for these leaves, we classify what is green and non-green. And with this, we can have this quite clean senescent dynamics as a percentage of the foliar area. Another example of applications of multi-class models, segmentation models, is the separation between different crops within one image, where we, in this case, monitor the growth of the different components of the mixed crops. So that's not limitative, but that's some examples. Voilà. So what we can say now is that semantic segmentation using deep learning algorithm is widely used for many purposes. It has replaced, in our case, all pixel-based algorithms for high-resolution images. Most of our models are usually two classes models. So one is, for instance, with heads and what is the rest, etc. But we want to go to multi-class models, which directly predicts, which identify which are the different classes, because it's known as more efficient and more efficient and with higher performance. But it's not so easy to train, because in this case you have to annotate each part of the image in different classes, so it's more work again. So one option is to use weak learning, where you can start from different binary models, which will provide you width, classification, with heads, leaves, stems, etc. And using this composite image, you can train a multi-class model. So it's also work in progress. And as a perspective, again, we think that, as presented by Etienne this morning, the constitution of large and diversified data sets and standardized is important for model assessments and improvements. So again, our large data set should be published this year by Simon and our colleagues. So with yes, different images and two classes, and it can be complemented with other data sets. And with respect to that, you are all welcome to contribute to build this kind of reference data set. What we can see as well is that complex covers. So, at least at Arvalis, in Arvalis perspective, it's less and less common to have pure wheat in the field or pure maize. We will have some weeds, and we will have maybe two crops at the same time. And so we need more complex models that will be able to separate the different components of the crop. So, and it will be more and more, it's more and more common in agroecology perspective. And the last, yes, okay, yes, so the conclusion was that phenotyping cultivars adapted for this complex system will require complex segmentation models. Voilà, but we are well, I think we are in the line to solve this complexities. And that's all. So thank you, everybody, and thank you for all the labelers who manually annotated the images. So from INRAE, Arvalis, and partners. Thank you.
Thank you, Benoit. Any questions to Benoit or the labelers? François.
About your last point, the complex canopies. So usually the example I see is a nice monocot and a nice dicot. Now, if we begin to have different varieties of wheat, is there any prospect that we can recognize them? Or if we have wheat and any grass, so it's more complex.
Yes.
So we will succeed, or perhaps not.
Maybe, well, there may be several ways to answer or to solve this complex question. We are going step by step, as you have seen. We are going more in depth at each time. So for now, we only consider, let's say, yes, monocots and dicots, let's say, or cereals and others. Maybe then we can go further. For this, we can also add, and we can reintroduce multispectral information as well, because it can be integrated in the same pipeline. So why not? But it's, I mean, if visually you cannot differentiate them, it will be hard to train models, and therefore it will probably hard to automatize this task, I would say that. And then maybe sometimes you do not need to consider only the very local parts, let's say the leaf shape, but also the position, so also more context, as shown by Tony for his, tomography problems. He has to consider larger images.
Thank you, Benoit. Any other questions to Benoit in the back? Yes, thank you.
Now you hear me? Yes. Thanks. I just want to see your personal preference. If you have to choose between semantic segmentation and can-I, so what will be your reply?
Again, sorry? It's a bit
if you have to choose between semantic segmentation and can-I,
yes.
So what will be your personal preference?
No, for me, semantic segmentation is the only way.
Take care, Marie is there.
Okay. Benoit can segmentation. Other questions? If not, I hand over to Benoit, I think.
Yeah, thank you. Thank you, Onno, and I will give you the hand for your presentation on high-throughput plant phenotyping of photosynthesis using the Light Induced Fluorescence Transient methods.
Thank you, Benoit. It's like we're playing a game here. Like,
yes,
but okay.
No, very good point.
Sometimes it's also, I'm Onno Müller from Forschungscentrum Jülich, where they say we have a lot of toys. But I would like to talk with you about high-throughput plant phenotyping of photosynthesis in the field, using one method, the light-induced fluorescent transient. Why high throughput? We have addressed high throughput in several ways, but I would like just to emphasize again, it's the environment. Francois already addressed it. The dynamic environment changes in minutes, in seconds, in terms of fluorescence. To quantify this dynamic response of genotypes in a varying environment, that's also the high throughput that contributes to it. The genes, of course, is always addressed, but here the environment to quantify this dynamics is important. How to achieve this high throughput in the field? I wanted to take the entry point of technical parts while use an optimal positioning system. We summarized it with Merlijn in this publication. It basically, if you have to do it by hand, do it by hand. If you can do it from satellite, do it from satellite. And the whole range has passed this morning, more or less in between, that we could use. To move a little bit further into what we have in Forschungscentrum Jülich, because we haven't seen each other for some years, and you're still welcome to come. I have the pleasure to make use of these different flying objects, but also the ground, not Pestrisson, the ground-based gantry systems, now recently also electric, going through the fields in functional throughput. I put functional there as it doesn't make sense to go fast if it's not needed, and also at relevant time points. When are these traits most distinctive? In the morning, in midday, afternoon, or at which time of the year? Why photosynthesis? Well, you may ask in this audience, why is this question? It's such a basic trait, but nevertheless, if in this audience we talk about yield, and often we don't come to a strong relation between yield and photosynthesis. Nevertheless, I think the basics of photosynthesis, if we could quantify it, that would be nice, to say the least. Nice is careful. But how to do it? Capturing this leaf with gas exchange, the 2 talks before, getting the Rubisco activation states. It's slightly difficult in high throughput, I may say. It may be in lower throughput functional still. But to go really in more genotypes, more dynamic measurement of time, I think we could better make use of fluorescence as indicator, where light comes on the chloroplasts. That was this animation here. Whoop, where I should click. The reaction centers close, and the more reaction centers are closed, the more fluorescence comes in competition with photosynthesis. This we can use to quantify the photosynthetic efficiency. How to measure this photosynthesis? Well, I said already, let's use fluorescence, and I will not go in detail about all these machine, all these sensors that we have here. Just what I wanted to achieve with this image is that we can measure fluorescence, and it has been measured for decades, from leaf level, from canopy, plant level, to plots and for breeders, from drones nowadays. These are more recent developments, passive sun-induced fluorescence, from airplanes, also sun-induced fluorescence. And in a couple of years, there will be a dedicated satellite from ESA called the FLEX satellite. What's the difference between these methods, to address that slightly? Well, of course, also the area that we're measuring. We're measuring here at centimeters in the leaf, and the satellite, we may wonder if this is still phenotyping in the sense of identifying the different genotypes. Well, if farmers have different genotypes on the field, you may call it also phenotyping. But these are the last ones. They work with passive fluorescence. If the passive fluorescence signal, to understand that, may be more complex, although highly interesting, but the rest of the talk I would like to continue on using active fluorescence, where we can get an estimate of photosynthetic efficiency by using active light source. So for that I would like to use the Light Induced Fluorescence Transient method. Why? It's active, it's fast, and you can measure remotely and proximal. What it does, it uses a blue light to activate the photosystems, and with this blue light it activates the first Quinone acceptor, and it puts a high frequency of blue flashes, so quick that we cannot see it. But these blue flashes, you may be familiar with the PAM system, which saturates your photosystems. Here the blue flashes in very high frequency, they also subsaturate your electron, your first Quinone acceptor. And you get the maximum, and to that maximum you can fit a transient. And then we're back actually, or we're at the place of familiar terminology for quite some of you, where we can identify the maximum fluorescence, the minimal fluorescence, and for that you can do the photosynthetic efficiency. We can also look a little bit further in the reoxidation. How does the first Quinone acceptor reoxidizes, and the reactions to the plastic Quinone pool. For the photosynthesis people, this trace may be additional trace for phenotyping, but I think the most potential at the moment in phenotyping, having a remote sensor that can quantify photosynthetic efficiency, remote 60 centimeters from the canopy, in a high throughput, that I would like to tell you some examples about how we used it in different experiments. With this short introduction, I also think I kind of covered again the slide I've used for many years now. What do we see as the field phenotyping concept? Over the last talks, I think we think more or less all the same. I put some examples over here. What I will continue with is photosynthesis, using lift in different examples of positioning system, in a drought experiment, a breed phase experiment, and under different environment conditions. To start with the drought experiment. For drought, although, one too many. For drought, I may not have to move. I could stay in Germany this year, maybe again, but this time we moved to Maricopa in winter in the desert. And with the colleagues there, Roberto Tuberosa, the lead, they irrigated this field such that it would grow durum wheat, 252 varieties. And we pushed a bike with this lift sensor over it. We actually is Nicolas Zendonadi. And then when the wheats were before flowering, the durum wheat, they stopped irrigation. With the stop of irrigation, the soil volumetric water content decreased steeply. This was in the first year, and we were pretty enthusiastic about the results. I will show you later. So we also did the second year, but then they controlled a little better. The blue line, they irrigated one line of the varieties, and the yellow line, you see a decrease in soil volumetric water content. Much more moderate over time in the second year. It was not so hot, it was a bit humid. Maybe this irrigation and next will influence it. But today, I will show you the steep decrease in soil volumetric water content. How does it respond? What is the response to photosynthetic efficiency? Yep. Well, it may not surprise you, but photosynthetic efficiency decreased. Also, the reoxidation parameters decreased. Here on the right side, presented in relative changes. And when the permanent voting point is below, you basically see a decrease in photosynthetic efficiency. Noticeable, there is, at the onset of drought, directly there is a slight increase in photosynthetic efficiency. And what you can also see is a strong variation between these 252 varieties. Obviously, from the previous results, I think you came to this conclusion already. Soil volumetric water content can be indicated by the plant status based upon the photosynthetic efficiency, but also on the varioxidation states. Of course, in the early phases, you may wonder what is going on there. There are some other processes ongoing which we cannot explain thoroughly. The question behind it, of course, is about the genotypic site and the heritability over time. And this is the days after withholding water. The yellow line, soil volumetric water content, and the other 3 lines for each photosynthesis trait, the heritability. You see heritability over time is not constant, decreases. Here we have more confusing parameters, maybe more environmental impact. Not sure to answer that. Nicolas, you can look further in the publication. I would need also Nicolas for more in-depth discussions about that. But that's what I would like to leave you with one probe of drought sensing and photosynthesis phenotyping in the field. If this is only one, it's 2 weeks' time that we were there measuring, getting a feeling of what does photosynthesis do with drought. But we are looking at, if we now go into estimates, actually the challenging title that Francois put up, can we predict yield? And being ambitious and a plant ecophysiological background, we say, if we would know in detail the sum of the intercepted light, old equations, or also presented here, and the light going to photosynthesis, then it would be in theory possible to estimate the biomass. So we did think, well, with our automatic systems and our lift sensors, we actually did acquire, over the growing period, a lot of photosynthesis, and also a lot about these conversion factors. So these conversion factors into photosynthesis, actually, we used in a simple model, and see how far do we get to this total biomass. And this is then not one time point, but these are, as you saw here, in 3 tables. Beat Keller, at the moment in Switzerland, working together with Andreas Hund, who is also here today. We measured over time the photosynthesis rate, put it in cumulative rate, and the efficiencies. And when you accumulate this over time, then in specific fields, for specific conditions, so you see not all these data are pooled, but nevertheless we were enthusiastic that by using this independent data photosynthesis, they correlated to the biomass data of soybean and maize. Okay, it's a first finding. It's a in that direction. I mean, there's a lot to discuss about this, but the high correlation shown here, and just using the photosynthesis data and high throughput, I think is an interesting way forward. How do we continue with that? Or what would be next steps? Well, of course, we could look in different environments, and also being aware of environment is highly variable. I sometimes like to have a bit more control of it. So we looked also in a more controlled environment, if you may say so, in the field, where we only increased the CO2 to future conditions about 50 years later, 600 ppm. The system you may be familiar with, it's existing already for 20 or 30 years meanwhile. You have pipes, they blow in CO2 into the field. Or inside this ring, the CO2 concentrations are elevated. They are controlled from the middle of the ring by a sensor. Well, we have this toy, the gantry system, that we can measure over the fields without walking in, and also without pulling down these pipes that grow with the canopy. And again, here is our lift sensor hanging to measure. We did in typical German crops, winter wheat, also in soybean, not so typical for the region. So let me start with the soybean. Though not so typical for the region, this was a hot year, and we had increasingly high increase in yield on the elevated CO2 of about 46%. The question of this project was a collaborative project with the University of Porto, was about the quality of this grain yield. If you put more carbon in, automatically your quality kind of decreased, shown in this graph. Getting this more carbon in, for us, was the question, how is that related to photosynthesis? How does photosynthesis contribute to that? And bear with me for a little table, apologies for that, but it's all here summarized. Again, 2 measurements. Is that high throughput? I don't know, but where we did intense measurements with the Leica on photosynthesis, obviously photosynthesis on the high CO2 is higher because they were measured on the elevated CO2, 32% higher, about 30%. at 2 growth stages. And interestingly, the photosynthetic efficiency decreases more during the pot filling stage than at the vegetative stage. What the mechanisms are behind there, we would hypothesize that this is a feedback inhibition to sugar, where at the early stages the sugars are not so limited, and so you have a small reduction, and later stages, to get the sugars out of your system to the pots, this may be more limited, or these pots may already be filled. To really find this causal relationship is slightly different, but it still keeps bothering. How can we really prove it? We may need to harvest the leaves to get sugar, but we also said we will look more in time. So soybean being one funding-dependent project. Having another project, we continued in winter wheat, and we say 10 varieties listed down here, and we grew them for the whole year. Again, basic data of biomass increase, about 20% increase in biomass. The yield increased about 5%, some limitations there apparently. And this experiment we repeated. This is actually, if you're interested to follow this experiment, the Phenocam breed phase, some publicly available cameras. You can see yesterday they were measuring in the field as we were talking, or as we were arriving here. Okay, this is just straw biomass, where I would like to go, of course, in photosynthesis. Do we also find, or what is the pattern during this growing period for wheat in photosynthetic efficiencies? Can it tell us a bit more about this sugar loading hypothesis? And again, a table over the year, the growing year, we measured about bi-weekly intervals. And what you see that photosynthetic efficiency actually, as I showed for photosynthesis gas exchange, is higher in the early parts of the year, when you have more growth, more CO2 available. And later in the year, you see the opposite pattern. This may be, that's also why these pictures, coloring, what we observed, there was earlier senescence later in the year under the elevated CO2 plots. The plants completed their growth cycle a little bit earlier, and this may be related to the lower photosynthetic efficiency. So a detailed analysis of photosynthesis trait. Now to connect this to the genotypes, we only have 10. So it's just poking a little bit in these mechanisms, what may happen, how it may be related to the genotypes. I hope to talk about that in another time, where we really look at the genotypes. The breeders already indicated, oh, that has this genotype, and blah, 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 it has this genetic information in it. But that I cannot talk about today. What I would like to end this is, as I say, we haven't seen each other for quite a while. Update what, besides the project I just listed, but we're still doing, is that we would also use a lift sensor for biotic stress in sugar beet phenotypes. If you have a colleague or a student in your group who will be interested in working with these photosynthesis sensors, we have now a PhD position open, and please feel free to apply or let him or her know. Looking forward to that. It will be large-scale breeders' experiments using the lift to detect mostly Cercospora, the black spot disease in it. Other part that I would like to share, and maybe I could present to you another time, is this photosynthetic efficiencies not only measured in CO2-controlled environment, but also light-controlled environment. Why light? Well, recently we completed agricultural photovoltaics. That is just solar panels on legs. And I had the pleasure to also be able to put a rail system under that, where I can measure the photosynthetic response in the shade and in the light. And of course, the final question is, can we have optimized crops under these panels that may benefit actually from the shade? For me, the question at the moment is to use this as a shade treatment on the light and photosynthesis, and shade transitions, light, quick sunlight on again. How does the photosynthetic response react? A big project in the bioeconomic sphere. With that, I would like to thank you for your attention, and before that, of course, thank the whole group of people I have the pleasure to work with. Don't think I did all this work. It's really a lot of people in our group that, together with Urash and Shizuo Matsubura, I lead together. And that should have been the last slide. Yep, and open for any questions while you can see some of these positioning systems moving. Thank you.
Thank you, Onno. So time for questions. Yes, one.
Thanks very much, Onno, for this very interesting system. My question is about the stability of the measurement across the day, during the day. How much does it vary with local environmental factors?
Directly. It's dependent on light. So it's directly related to light, the photosynthesis, but you can control for that. That's one of this Durham-Reed experiment. I didn't show it, but that's where we included light as a variable. Because we couldn't analyze these 252 genotypes in 1 hour or a few seconds. So we had the variation of light, environment, temperature, humidity over the day, and it varies. Yeah.
Okay, and so can you take these factors into account to correct for?
Yeah, no, that's this phenotype, that is the model. We take them into account as cofactors. We have to, I would say. Yeah? You cannot ignore them.
Okay.
And with that, there's still enough, what you saw here, information left in the photosynthetic efficiency.
Okay.
Yeah.
Okay. Thanks very much.
Yeah.
Yes. Just one question on the go. Do you make measurements during the night as well sometimes? Or?
Sometimes, usually my short part in when to measure, there were some night measurements also, I think. The nice thing, or maybe this sensor is really working well on the daylight and capturing these variations of the effect of the environment during the day. During night, photosynthetic efficiency is slightly boring in terms of if they are not severe stresses, it's 80% is 0.8. So yeah, there may be difference in the severe stresses in different varieties, but yeah, mostly we measure during the day.
Thanks for your talk. My question is about like using fluorescence. If you think about breeding, and for instance, you get a good heritability, a good relation to yield or something, you can go ahead with that. But if you think of modeling yield, as you said before, probably you need a kind of validation for that with other biological process. Did you do anything on this issue, or do you plan on doing something on this?
Yeah. Well, that's the slide. Why photosynthesis? Eh? If we have to have a discussion, if that's not a biological process, and the fluorescence coming from that, then it's complex. But because that's a really nice thing, elegant thing about fluorescence. I cannot say it differently, that it is a biological process directly coming when there's light on a leaf. The light is transported in the chloroplast. This is a biological process. I mean, it's measured physically, but there is not much. There are all the validations also to what I said earlier. Maybe the photosynthetic efficiency or the fluorescence use is an old method, and why were there people so enthusiastic about it? Also, they compared it to the gas exchange and to all this validation, so that it is really reflecting the electron transport rate processes. Yeah.
Do you think like this will need to be a kind of link to carbon assimilation and these things to the parameter then be?
Yeah, that's the electron transport rate multiplied by the light intensity. That is the input of the energy for the carboxylation. The only missing part there would be, not only, but one of the missing parts there is the respiration that you have to account for. And that is still an unknown that you cannot measure with fluorescence. But you have your incoming energy quantified.
And you think linking the high throughput with other methods that we already know is the way to do that?
It's an interesting way. I mean, I'm open for a lot of, and it's certainly, I think structure and function, I said for our group. I just focus now on photosynthesis, but I certainly don't claim that photosynthesis is the only answer. It's a combination of structure and other functions. Yeah.
So if you consider the difference between different genotypes, how important do you consider the canopy coverage and the leaf orientation as a confounding factor for your trait?
Very important, actually. Yeah, I cannot. I didn't. I mean, which model? Where did we do it? Yeah, if you would like to have numbers, but yeah, very important. That's the only thing I can say. Yeah.
Are you going to measure that? So is it kind of factored in somehow, or you just are you going to measure these?
I mean, it's a little bit a step further. The elegance of this photosynthetic efficiency is that it's a relative signal on the leaf. I also addressed a little bit in the beginning sun-induced fluorescence, which is image-based and covers the whole canopy. And there we still have to use, if you have more chlorophyll, you have more fluorescence, and you cannot directly calculate with it as an efficiency. And in these aspects, really the canopy structures become really important to get a realistic photosynthesis estimate of your canopy in sun-induced fluorescence, because it's not only green value, it's also the leaf angles and the transmission of light in the canopy. So at that point, it really becomes important, the intercepted light. In this relative method, it's actually less of an importance to get a good photosynthesis estimate.
Thank you, Onno, for your presentation and answers. We will move for the next, to the next presenter, so Sylvain Jay, which will talk about chlorophyll content estimation from multispectral and RGB images.
Leaf chlorophyll content estimation in wheat crops using two types of imagery, so RGB and multispectral sensor. So this is a work I conducted with the help of Frédéric Baret, Marie Weiss, and Frédéric Venot for the INRAE side, and Gaëtan Doubige and Benoit De Solan for the Arvalis side. So as you know, leaf chlorophyll content is a critical plant trait for crop phenotyping. For example, to study plant physiology or because of its relationship with the nitrogen content. And actually, close-range sensing is an interesting alternative to other way of measurements, such as chemical analysis, which are destructive, SPAD-like measurements, which are only punctual, or satellite imagery, which is too coarse to focus on the leaf part. So chlorophylls absorb lights within the visible range, which make potentially RGB and visible and near-infrared multi- or hyperspectral sensors adapted for LCC estimation. So, of course, many studies have demonstrated the interest of multispectral and to a lower extent RGB camera for LCC or nitrogen content retrieval. But actually, fewer studies have compared these two sensors on the same crops. So, just I have listed a few pros and cons for both types of imagery. So, in terms of spatial resolution, usually you have a better resolution, spatial resolution with RGB imagery. But of course, with multispectral imagery, we have better spectral resolution with bands that are narrower, whereas with RGB imagery you only have three bands, and the bands are wide. So, but on the other hand, the RGB cameras are of course less expensive and are often more easy to use. Compared to multispectral imagery that may require some additional pre-processing step, as Benoit showed this morning with, for example, some band registration step. So in this study, I will show a comparison of the performance between these two kinds of imagery for the estimation of image level LCC on wheat crops, so the average LCC in the image. And I will show you some very preliminary results on the estimation of LCC vertical distribution in the crop. So here is the outline of my presentation. I will first present the data. Then I will talk to you about the estimation of image level LCC, and I will move toward the estimation of LCC vertical distribution. So the data were acquired in 2019 in 3 sites in the south of France, and at 5 dates along the growing season. So you have some examples of images showing different soil background and illumination conditions. So basically, we try for each scene to acquire to image the scene at 2 times during the day to have different illumination condition. So this makes 67 images in total to evaluate the algorithm. So regarding the LCC measurement, so at first we wanted to estimate LCC at the pixel level. So we put some labels on the leaf, actually 30 labels on the leaf that you can see here with numbers ranging from 1 to 30. And we measure with a SPAD device the LCC for each of these 30 leaves. And then we average the LCC values to provide one LCC for each image. So regarding the RGB and multispectral imagery, we used, I would say, literal-like measurements, illustrated with Gaëtan here. So for RGB images were required with the Sony RX0 camera, with, of course, red, green, and blue white bands. The spatial resolution at the ground level was lower than 1 millimeter. Very importantly, we perform radiometric calibration to turn the value into reflectance value using the panels you see in the image here. And that's it for RGB imagery. In the case of multispectral imagery, we use an airfan camera with 6 bands ranging from the blue to the NIR spectral range. The spatial resolution was similar. We performed also a radiometric calibration, and we used a band registration algorithm that we developed in CAPTE. And this algorithm allows us not only to register the band, but also to provide a distance image that can be superimposed with the bands. So, as an example, I show you 2 images of the same scene provided with the 2 sensors. So you can see that the RGB image is actually, the RGB reflectance image is actually much more yellow than the true color image derived from the multispectral image. And this is actually because the red band in the RGB camera is actually less red than the red band in the reflectance image. Actually, we don't know exactly the spectral sensitivity of the camera, but we can assume that the red band is actually located between the green band and the red band. So, about the estimation of the estimation method in itself. So first, we need to, of course, to classify the green pixel, because we are interested in chlorophyll, so we want to first keep only the green pixels. So for multispectral imagery, we used the same methodology we used in a previous study for sugar beet crops, which is just to compute the vegetation index image and thresholding it. And with RGB imagery, we used the algorithm Benoit told you about earlier to segment the plant pixel, and then we keep only the green plant pixel using a similar method as before. So we basically use a data-driven approach for both imagery. So we have the images, pixel level reflectances, and we have some measurements for each image. So the first thing is to take the average of the measurement to have 1 measurement per image. And regarding the reflectances, we apply several mathematical transformation and so on. So first, we average the reflectance over a subset of pixels. So this subset can be all image pixel, or only the green pixels, or only the brightest green pixels, which are supposed to be less affected by the canopy structure, or in the case of the RGB camera, which has a very large field of view, we can limit the view zenith angle to 10 degrees to limit the bidirectional effect. Then once we have the reflectance for each image, we can apply some possible pre-processing. For example, we can subtract the blue band value to other bands to remove the influence, or at least to decrease the influence of specular reflections on the spectra. We can also apply some logarithm to try to decrease the nonlinearity due to the increasing saturation due to increasing LCC. Then we have some transform or untransform reflectance. Then we can add some other variables that can help handling with the difference in illumination between the images. So this can be the sun-zenith angle, this can be the relative solar row azimuth angle, or the diffuse fraction. And we can test every combination of input variables. And we do, so it makes about 63 combination maximum for the RGB camera and more than 500 combinations for the multispectral imagery. And then we apply a data-driven approach, a machine learning model, such as GPR in our case, to estimate the LCC. Importantly, we use cross-validation to assess the performance, so using n-1 sites for calibration, and the remaining one, which is independent, for the validation. So here are the results for multispectral camera. So here I plotted the error, the estimation error, for different subsets of pixels. So either all the image pixel, only the green pixel, or only the bright green pixels. So as expected, the best performance are obtained when focusing on the bright green pixels. And you can see here the results you can obtain by using all the just the reflectances, or just the reflectances and the angles. And the best results here are obtained by taking the best combination of variables when considering the transform reflectance, either by just applying a logarithm to the reflectance, or by subtracting the blue band to other bands and applying a logarithm. So we have quite good performance with less than 5 microgram per square centimeters. And then if we want to compare with the RGB, this is what we get. So in brief, the best results are obtained when restricting the field of view of the camera. Similarly, to have more or less the same field of view as the multispectral camera, focusing on the bright green pixel and using the same mathematical transformation applied to the spectra. So in this case, just the RGB bands. And which is interesting here is this is actually we have the same performance between the two cameras. So this partly answers to what Shawn says earlier in this presentation, is that a well-calibrated RGB camera is okay, actually, compared to multispectral camera. You can do a lot of things with RGB only. So I just plotted the estimated versus actual value. So you can see that they are pretty much the same. And in both cases, the best variables, the best set of variables were very similar in both cases. In the multispectral case, you use the green and red bands minus the blue bands plus a log transformation, whereas in the RGB case, you use the red band minus the blue bands plus a log transformation, and we use also the azimuth angle here. So just as a very preliminary results about the estimation of LCC vertical distribution. So how can we do that? Actually, we can exploit the distance image we have from the band registration. So here you have an example of a true color composite image derived from the multispectral image after registration. And here you have the distance image you can obtain in centimeter. And then you can focus on some specific layer in the canopy. Here, by I extracted by a 10 centimeters wide layer. So you can see that you go deep into the canopy until the soil background. So this is how we can basically do that. And here are some very preliminary results about the estimation for each layer. So we build a model for each layer because for each layer, the best combination of bands differs. Typically, the red edge band and NIR band are more useful for the upper layer, whereas in the when you go deep into the canopy, I guess this is because of maybe multiple scattering, the RGB bands are more helpful. And if you have a look at the best combination for the four layer, in this case you don't use the red edge band or the NIR bands at all. And this is why when you focus only on the RGB bands, you have some kind of similar results, except for the upper layer, where the red edge band can help to improve the estimation. So in brief, I would say that in the specific case of LCCM estimation on wheat crops, multispectral and RGB camera are obtained about the same performance. And we have very promising results for the estimation of LCC vertical distribution, actually a bit far beyond my expectation. I didn't expect to estimate chlorophyll content in deeper layer with such a good accuracy, but okay. So that's it. Thank you.
Thank you, Sylvain. So one question, Pierre.
Thank you very much. That was very interesting. I have one question, or several in one maybe. If I understood well, you use as a measurement of chlorophyll content actually the SPAD measurements. Is that correct? Yeah. Yeah. But the SPAD is not. Well, the calibration of a SPAD will depend on is variety dependent, and also it's not linear through the whole range of chlorophyll content. And you said that you were more interested in nitrogen, so I was wondering why you didn't measure directly nitrogen content in the leaf. No. And the last one was, it may be related to that, that's why I said it's only one question, because I saw that the difference through the canopy, you have a decrease, the extension of nitrogen through the canopy is pretty low. You still have a lot of, although in your image, your canopy seems to be very, well developed. And so usually the content of nitrogen in the canopy decreases exponentially and following more or less the light. So I was wondering also if you plot it against the light, actually, the fraction of FE par.
Which one? This one?
Here. Yeah, if you have the same scale, which I think you have in different plots.
For the different?
Yeah,
but they are not necessarily on the same plants. Huh?
What do you mean?
I mean, these points are not obtained on the same plant as these points, necessarily. Yeah,
but still, you expect to have very little nitrogen at the bottom of a crop. Usually you have if the canopy is closed, the SNN at the bottom will be like 0.3, which is about the compensation point for photosynthesis. But we can discuss that. But yeah, I was wondering, there's no nitrogen gradient in your canopy here, basically, which is very weird. But I wonder if it's because the correlation between nitrogen content and SPAD measurements is actually a logarithm.
Actually, in this case, I'm not interested in nitrogen. I just put the word nitrogen once at the beginning because it is usually considered as correlated with chlorophyll, but I'm not interested in nitrogen here.
Yeah, but chlorophyll and nitrogen in wheat usually pretty well correlated. So,
yeah, actually, I think it depends on the varieties and the stages. And for your remark about the SPAD measurement, actually, we take into account this saturation with when converting SPAD measurements into LCC measurements.
Thank you. More questions? Yes.
Thank you. The last point in the comparison between multispectral and RGB with the decreasing prices for multispectral cameras, why at a certain moment do we still have to ask that question? I mean, if you can use multispectral sensors for the same price, for the same resolution, and knowing that it's related, they can have wavelengths at the chlorophyll bands, why we shouldn't use multispectral?
Yeah, but I'm coming from the hyperspectral world, so I'm not promoting RGB camera. I'm just saying that. in this case, we obtain the same performance with with the 2 cameras, but I don't say that you can't do more with multispectral camera, such as retrieving other pigments or many other things like any other biochemical properties.
Yeah. Thanks, Sylvain.
Okay, thank you. Next time you will show results with monochromatic, with monochrome cameras. So we will move to the next talk by Onno. So we finished with the technical talks for today, and we will have 2 more talks and then some information. So one talk by Onno on an emphasis. No, it's technical as well? Yeah. No, I don't know.
What's another talk?
No, the other one is by no. The other one is by Fred. So I think you have one on emphasis.
Yeah, I do. Sorry to bother you again, but actually the title says it's emphasis on opportunities and demands for field phenotyping. And I was asked by Roland to give this talk, and the rest of the talk will be also a Roland talk. I try to imitate him, but why I said yes to that is the example below here. I called it a toy that I have the pleasure to play with. I may have designed it, I may have thought about it. I think it's not mine. It should be shared. It's an infrastructure. A lot of money went into it, and actually through the access of EPPN, many people worked in this project and could work in this project. So it was an infrastructure that was available, is available for many people, and I had the pleasure to work together with a lot of different scientists, from entomologists to also people looking at the moment at leaf structures in detail on the elevated CO2. So for me, that is the exciting part of working in such a big infrastructure. And it was addressed already today. What is the validity of maybe working only one place of a reasonable good agricultural region in Germany, but wouldn't it be much nicer to compare this to a dry region in Spain and other parts? And that's why I said, okay, Roland, you cannot be there, you have another conference, it's okay. And then the organizers kindly put my call talks right after each other, but okay, that's, I leave up to the organizers. So bear with me, but I'd like to introduce you a little bit the emphasis project. Larger, indeed not so much technical, Benoit, a larger mission. There are many candidates here in the room who could maybe give a much better talk than I, and are open also for questions. I'm looking at some persons in the organization here. But emphasis, a European infrastructure for multi-scale plant phenotyping and simulation. You see already the ambition for food security in not a changing, but a changing climate. The same background information, we have the environment and the genes. And together, if we do this phenotyping, we think we can solve the challenges. Highly pitched, but plant performance and plant production, we do think, and I hope we all think all together, that higher quantity and quality of biomass could be achieved by all these phenotyping actions we do. We could identify novel characteristics and products. We saw some today. Yielding in stressful environments, and sustainable production intensification. We had some agroecology today. Sustainability was addressed a little bit. So we're all working in these bigger aims. And as such, also, we come from the background, I think there's no doubt, there is a demand for plant phenotyping as a tool in different environments, in specialized infrastructures. It's not only the infrastructure, not only the trait detection itself, also the modeling. Nice examples of that also today. And the data reusability, FAIR was already mentioned, integrated approaches across sites. And actually we're doing a pretty good job in Europe in having all these infrastructures and sitting here all together also in France. We were actually also, if you often compare us to the United States, we were the global leader. Australia did a lot also, I must say, but now competition is growing. To pitch this for Europe, I think we're still pretty good. It came from a background that a lot of national infrastructures were developed. In Germany, one of the early ones was the DPPN. At the same time, slightly earlier, was in Australia, the Australian Plant Phenotypic Network. Not much of Europe actually there, but okay. Then there was Phenom in France, Ireland. You had a talk today about NPEC in the Netherlands, not on this list yet. It's just will open in September at the IPPN conference. And also project, regional or European project networks elevated. I mentioned already the EPPN that I favored from personally in the breed phase experiment, having many researchers coming there in the field experiment. Continuing this, or bring this together, making use of these infrastructures across Europe in different regional projects, that's actually what Emphasis has in mind. To create a long-term stable organization, and if some of you may be familiar with the term ESRI, European Support for Research Infrastructure, that is where we would like to put this plant phenotyping infrastructure that we have. Don't see it as single entities in all different countries, but you see it as a whole European infrastructure. And for that, so that is here is the fancy slide, what I just said. The objective is to develop an infrastructure and providing access across Europe, European data level, and develop and evaluate, share knowledge and novel technologies. The nice words on this. What did we do for that? How did we get to this road to operation? Well, we, Roland and colleagues, it started several years ago. Just the preparatory phase ended last year. And meanwhile, the implementation phase has started to get Emphasis really as an infrastructure project, an S3 project, which should lead to an operational phase in 2023, where all these different national infrastructures are to be connected and that we have full access to them. How does that look like at the moment? Emphasis being a phenotyping infrastructure, not only field phenotyping, but in different infrastructure categories. It was divided up in the basic categories. We had some of them today also referencing to controlled environments all the way to data collection. I will shortly go through that. The controlled environment, already addressed by Francois today, bringing the greenhouses and growth chambers, simulation and monitoring of the environment. High level of automation, we saw that in the talk of Rick also, and in the talk of François a little bit. Deep phenotyping, throughput typically about 100 to 1,000 plants. And we have a lot of these installations, 112 in 19 countries over Europe. Some examples shown here in pictures. Other category that was defined were the lean fields. Environmental monitoring should be in the field. There is equipment for phenotyping basic traits. There could be ground-based or airborne sensing going over the fields. And it's a clear network of fields. You could think of the breeder fields that they use already to test their new genotypes. They already actually have a setup of fields. The drops, or the other project mentioned earlier, also one of the examples where lean fields were used. Also a considerable amount of sites in 14 countries across Europe, lean and intense. Intense field, I gave you an example of the breed phase as one of the intense fields. Has to deal with environment monitoring. High quality is also a term, but okay. High quality measurements and detailed measurements, and field sites enabling environment simulation. Same number here because we pulled lean and intense fields together. Here you see Malcolm's bridge, the lambda tech system earlier. Well, we have seen actually, all, except this is in Belgium, the examples we've seen today. Going somewhere with all this data, we obviously need the modeling. So virtual platforms, and we put it in a separate category. With virtual platforms, different types of models, crop models, and integrated interfacing with different phenotyping installations. I must say, well, that I'm now not sure which this example that I didn't look up is from, but as you not surprising, we have different models, also a high number, for plant phenotyping available. Some new models were introduced today also that we can make use of, so maybe that number should increase again. Next to the models and getting all the data together, we have the different information systems being made use of, based upon the European FAIR principle of sharing and having access to the data, and the statement, but the emphasis that we should all have an integrated information system. Some of you in France, they don't know, use the Phenom. We have the FIS system in Belgium, or PIPA in Belgium, if I'm correct. The other one, it's not on there. Yeah. So these are the different information systems, the different categories, And now I'm back to actually why I said I accepted it, why I think also this whole emphasis project, I'm very happy that Roland does all this work, because the previous work that Roland was also involved, having the EPPM 2020, this really enabled a lot of access to the facilities, but also a lot of connection between scientists, and I don't say automatically following our success rates of having publications, how we are evaluated often by publications. I don't say that's only a success of a scientist, by the way. But to continue that emphasis is one whole European initiative that can continue this access. So with that, it provides a sustainable service for the whole European community, with access for the users, industry engagement, communication, advancing phenotyping practices, data management and modeling, and training and education. A lot of buzzwords. What does it mean? These are my words, sorry about that. What does it mean in practice? Well, emphasis contributed significantly to getting new research infrastructure projects that have funding to provide access to these different installations that are available in emphasis. 3 examples that some of you also work together, a lot of you actually work on. Aquasurf will start in 2022, a bit agricultural research, access to the facilities with their background. Infrastructure for artificial intelligence, AI for life, also starting in September 2022. And now being just submitted, the VNet, developing next generation information methods in also agroecology. So infrastructure projects with access, available through the emphasis, or also with emphasis. And with that, that is the last non-technical emphasis talk I was happy to give you on behalf of Roland. I could try to answer any questions or forward the questions to other people in the audience. Thank you. Who was not aware of emphasis? Just, so I was just telling it for nothing. Okay, sorry about that. I didn't see any raised hands.
Okay. Thank you. Okay, so we end the formal presentation, and we have now some announcements. So one from Fred.
Okay, so it's again a non-technical speech, but short. So as an editor-in-chief of Plant Phenomics, I wanted just, because Plant Phenomics also is sponsoring this workshop, I wanted to take the opportunity to provide you some information about the journal. So it's a journal that is published under the SPG Science Partner Journal of the American Association of Advances in Sciences. The scope is plant phenomics, which is at the crossing of different disciplines. So okay, now you have got a better feeling of what is plant phenomics. So there is quite a large scope of manuscript that can be submitted and accepted by plant phenomics. There are different categories of articles. Can be just research articles, but also reviews. It can be also data papers. It can be a publication of methods that are well accepted by the community. It can be opinion papers, short ones. These are the editor-in-chief, so Seishi and Zhongming Cheng and myself. And then, of course, we beneficiate from several associate editors. So this is the list, and it has changed a little bit, but not that much up to now, from different countries. Just some statistics. So the journal was started in 2019, and you see the increase in terms of number of articles and number of citations coming from different places.
in terms of number of articles and citations. And then, this is maybe the information that you are looking for. The impact factor, so we don't have it yet, but we should have it next June 2022. And it's expected to be, I don't know, but I will give you some. I'm not allowed to say what it would be, but okay, I've got some tables. So in the plant sciences category, it should be about there, so which is not too bad, not too bad. For the agronomy, it should be there, which is very good. And for remote sensing, it should be there, which is not too bad as well. So now I think you are very motivated to submit manuscript to Plant Phenomics. Here, where the place where you can do that. And of course, because I guess we will receive quite a lot of manuscripts from this conference, and of course, so you are very welcome to submit your manuscript regarding the presentation that you made or from other studies. So very shortly we will need reviewers. So when you receive some invitation to review, you could do, because it's a very good journal, so it was to do it. And probably we will need also additional associate editors, so maybe some of you will be contacted to be part of the editorial board. I think that's it, so thanks very much.